Hello dears, good morning. Welcome to our program NEET Foundation, and today we are going to discuss about the concept cell division and cell cycle. So tell me, what is about the cell division and cell cycle? What is cell first of all? Cell is a basic unit of life. So within that basic unit of life, continuation of life, the cell plays a very major role in continuation of life process. Continuation in the sense it has to grow, it has to die, and after death again the regeneration of cells. If we get any damage of our skin cells or any sort of organisms get damaged or injuries, so the repairing worn out cells will be repaired by the cell division process. It might be of mitotic cell division process or the meiotic cell division process. So let us see the introduction part. The cell division is considered as a fundamental characteristic of an organism. It's a fundamental basic unit. Whatever the life processes do occur in the living organisms, basically it occurs in the cell, right? So within this, it results in increasing the size of each organism. But immediately before beginning the process of cell division process, first the cell will be showing the process called as a anabolism process. Anabolism in the sense, so it goes the protein synthesis process. So as the protein gets into the nucleus, chromosomes and all, so the cell gets the capacity to divide itself whenever there is a requirement. Okay, so see here the picture indicates that this is a cell anabolism process, nothing but the protein synthesis. Okay, so cell, they are getting ready themselves for division process. Okay, so here the cell shows anabolism and accumulation of cytoplasm. Each and every cell, whether the animal cell or the plant cell, there we can observe the suspension of cell organelles can be seen inside the cytoplasm. Cytoplasm is a jelly like structure which is present inside the cell. Okay, so here the accumulation of the all together, the cytoplasm gets together, accumulates and at the maximum rate. So here we can observe the cell division is responsible. So here the cell is getting ready for the division process by the protein synthesis. And what is the importance of the cell division? It helps in continuing the race of all the organisms. So the ecosystem will be balanced. Okay. Next. So next let us go the other topic. So who discovered the cell division? Right. So in the year of 1850, the cell division was discovered by the scientist von Mohl, and he told about that at the same time it was been another scientist. So Virchow he told that the pre-existing cells are responsible for formation of the new cells. First division of the cell division was discovered by the scientist von Mohl. And later on cell division, after the cell completion, division completion process, what happens? New cell forms. New cell, how it forms? Based on the previous cell's existence. If they exist and undergo their own metabolical activities inside them, then they can have the capacity to produce the new cells. That was being given by the scientist Virchow. And who is another scientist here? Wilhelm Hofmeister. So here he told that the cell divides. So here the nucleus is dividing into daughter nuclei. Single nucleus is dividing into two nuclei, which is followed by the division of a cytoplasm. First the division of nucleus occurs, next we can observe the cytoplasm divides and completely dividing the single cell into two cells. So here we can observe single will be dividing into two cells divides and form 4 cells. 4 cells divides and form 8 cells. 8 cells divides and forms 16 cells. Okay, so in this process, so these are the different types of scientists, different scientists involved in the invention of the cell division process. What happens actually, and internally keen observation was being done by him by the cell division. Nucleus divides and then we can observe the division of a cytoplasm. And what is the next? Yes, what is the importance of the cell cycle? Okay, so cell division and importance. What are the causes? Why does it causes? When do we can observe the cell divides? Okay, so here the cell is dividing. Damage or any wound in the body, as we discussed previously, the damage, if we get any injury, after that we will be getting the healing of the wound. Okay, so wound forms. As it dries, immediately we can observe it will be peeled off. So after peeling off, if you remove that peel off of the wound, we can observe the skin will be regenerated, right? So that is by the cell division process. Okay, and damage or any wound in the body of an organism causes increasing the rate of the cell division. Okay, so when does it occur? When there is a damage of the cells or damage of the any organ, we can observe the increasing rate of the cell division after the wound forms. So initiating the growth and differentiation. Okay, so during the development of baby, so after the reproduction process, after fertilization, it forms the zygote. Okay, so what is a zygote? It's a diploid nuclei cell. Okay, so there we can observe the nucleus will be dividing and forms the embryo. Different types of cells will be formed in the embryo in the multicellular organisms, and there we can observe the formation of cells will be undergoing the process of mitotic cell division, and then the cells will be forming as a tissues together forms as a organs completely divides and forms, together forms the organ systems, and the multiple organ systems will be formed in the mother womb. Okay, so the zygote, after the fertilization process, sexual reproduction, the fertilization process takes place. After the fertilization, we can observe the formation of zygote, develops into embryo, embryo develops into fetus, fetus develops into baby. So during the initiation and differentiation, initiation in the sense different types of cells will be initiated. First they will be formed by the zygote. Okay, so in the embryo multiple number of cells will be seen, and in the fetus we can observe multiple formation of the organs by the mitotic cell division. Okay, so differentiation of the body organs, organ systems, and complete organism is being formed by the help of mitotic cell division. And what is the next process? Development of different organs. As I told just now, each and every living organism which is made by multiple cells. We have muscle cells, we have nerve cells, we have bone cells, we have another cell, skin cells. Yes, so all these different types of cells are being formed during the embryonic stage, initiation stage, that grows and different organs will be formed. Organs together form the organ systems all together club and form as a living organism, right? Next, our next concept is different types of cell divisions. What are those? First one is the amitosis. What is amitosis? It is different from the mitotic cell division, and it is called as a direct cell division. In the mitosis we can observe the stages, phases, nothing but the prophase, metaphase, anaphase and telophase. There we can observe the duplication and recombination and synthesis of DNA for the formation of the multiple n number of chromosomes. Okay, but coming to amitosis, it occurs in the bacteria, yeast and amoeba and the tapetal cells. It is primitive cell division. Why it is called as a primitive cell division? Time duration, it takes very less time, and here we can observe the parental cell karyokinesis, division of the nucleus, and here we can observe the cytoplasm, division of the cytoplasm, nothing but the cytokinesis, and then we can observe the single parent will be dividing into two individuals. Okay, so here we can observe the cell partition. How many cells will be formed? Two. One will be dividing into two individuals. Okay, so this is the direct division, mitotic cell division called as a amitosis. So what is the benefit of this? So time duration will be less, exact copy of the parental cell will be formed as a daughter cell, right? So there is no formation of the spindle apparatus. What is the use of spindle apparatus? So those spindle apparatus are responsible for just holding up the chromosomes which are ready for dividing into a new cells. But coming to this, there is no division of the chromosomes. Okay, so directly the nucleus will be dividing and forming a equal distribution of the nucleoplasm and the genetic material which is present in this. So there is no involvement of different phases which occurs in the mitosis. Okay, And next one we can observe the karyokinesis, division of nucleus, cytokinesis, nothing but the division of cytoplasm, and complete two individuals will be formed in the mitotic cell division. Hope you all understood. Next concept is type 1 is the amitosis, and type 2 type of cell division is called as a second type of cell division is called as a mitosis, also called as equational or homotypic division. The word itself indicate equational. What is equation? Equation in the sense will have left hand side and the right hand side. Reactants will be reacting with each other and forming a product with a same number of molecules in the chemical. Yes. So, here also we can observe the division of mother cell, which is segregating, distributing its own chromosome into its daughter cell. Okay. So, equational, number of chromosomes which are maintained by the parent cell also will be maintaining by the daughter cell. That is nothing but the equational division. And also homotypic division. Homo, the word itself indicates it is a same. The qualities, the characters, the traits which are present inside the parent, same kind of characters will be segregated towards to the, means characters will be passed towards to the offsprings. That is the reason the mitosis is considered as a equational and homotypic division. Okay, so who invented this? Mitotic cell division was being invented by Walter Fleming. Let us see into this. The term mitosis was given by Fleming in the year of 1880, and it occurs in vegetative or somatic cells. What are vegetative cell and the somatic cells? A very easy process we can keep as a blind code. Somatic cells and the vegetative. Vegetative in the cells, normal cells apart from the reproductive cells. Okay. So, there in the cell also we can observe two types of chromosomes. One is the autosomes and one more is the allosomes. Autosomes will be forming the somatic cells by undergoing the process of mitotic cell division, and next one is the allosomes, also called as a sex chromosomes. So, these are responsible for formation of a gametes, and further division will be undergoing by the mitotic cell division. Okay, the gametes are being formed by the third kind of cell division called as a meiosis. That we will be discussing in the next class. Okay, and it occurs in the vegetative or the somatic cells. So, in our body, what are the somatic cells? Brain cells, skin cells, other vital organs, heart, lungs, liver, kidneys. Apart from reproductive organs, remaining vital organs, everything comes under the somatic organs. Okay, so these are been organs are been formed by the somatic cells, done by the type of chromosomes called as a autosomes, which undergo the division called as a mitotic cell division. And it occurs in both diploid and haploid cells. Diploid is nothing but after the formation of zygote. Zygote is a diploid in condition. It forms as a new individual in the mother's body, and in the haploid cells first of all it forms as a gametes, and as it grows in the embryonic condition, the reproductive system of the baby will be formed first of all by the meiosis, and it is followed by the mitotic cell division for its growth of the organ system. Okay, and two daughter cells are formed at the end. Which contains equal number of chromosomes. Okay, and it is also called as duplication division, nothing but exact Xerox copy of the parental cell. The cytoplasm Xerox copy, the nucleus Xerox copy, the type of chromosomes Xerox copy, number of chromosomes will be Xerox copy. So, it is called as a duplication division, or also called as equational division. Equal number of cells, chromosomes and type of cell organelles will be seen in both parental cell and also the daughter cell. Hope you all understood. Coming to the next concept, let us see the stages of the mitosis. Before beginning the stage of mitosis, the first stage is called as, initial stage is called as a interphase. What is interphase? Interphase is nothing but, so here we can observe the different type of stages. Okay, so first stage is the G1 phase, next stage is the S phase, and next phase is the G2 phase. and after G2 phase we can observe again division of the mitotic cell, division of a formed daughter cells. So it is subdivided into 3 stages. How many stages? G1, S and G2. So these plays a very major role in making the chromosomes to get ready for the cell division, okay, and replication or duplication of DNA occurs in the S phase. Okay, so what is the G1 phase? G1 phase is the ending stage of first mitotic cell division from the parental cell and beginning of the DNA replication. DNA makes the chromosomes to get divided and equal amount of chemical substances, the base pairs will be equally distributed towards to parent cell and also towards to the daughter cell. Okay, so it opens and distributes its chemicals towards to the cells, and this synthesis phase, synthesis of the S phase, nothing but DNA opens. The DNA will be in a double helical structure. It opens and releases the chemicals for making the chromosomes to get ready for the division process, and again it closes. The double helical structure opens and closes. This is nothing but the synthesis phase or the replication phase. And next, synthesis of protein and RNA occurs in both G1 and the G2 phase. What happens? Synthesis of proteins and RNA. What is the use of protein? Makes the cell to get energy for the division process. What is the use of RNA? It's also one of the proteinaceous molecule which is also helpful for division process. Let us see the picture over here. So here we can observe the cell will be formed. So within the cell we can observe the centriole and the formation of microtubules making the cell to get ready for the cell division process. The nucleus will be formed and the chromatin. Here we can observe the condensed shape of the chromosomes. Okay, so they are getting ready for division process which is being done by the duplication and replication of DNA, right? Next stage is the mitotic phase. Okay, so within the interphase we can observe first process is called as a karyokinesis, nothing but the division of nucleus. Okay, so single nucleus will be dividing into 2 nuclei. Okay, so karyon means division of the nucleus. Okay, so karyokinesis is the division of nucleus, and after this the 4 phases, the prophase, metaphase, anaphase and telophase. After telophase we can observe the division of a cytoplasm. Cytokinesis, nothing but division of the cells. Division of the cells by the division of cytoplasm. Before to that we can observe the karyokinesis first of all, before the beginning of a mitotic cell division, and during the ending stage after the 4th phase of the mitotic cell division it undergoes the cytokinesis, nothing but the division of cytoplasm. Okay, so that is nothing but the karyokinesis and cytokinesis. Let us see the first phase, prophase. The diagram indicates here we can observe the chromosome structures will be condensed inside the nucleus, and here slowly the nuclear envelope, nuclear membrane will be degenerated, and here the chromosomes will be coming in contact with the microtubules, nothing but the spindle fibers. So they are helpful for suspending freely chromosomes to attach towards to those for the division process. During this phase the replicated chromosome each consists of 2 sister chromatids. Okay, so the chromosome will be divided into 2 chromatids in the upper region P arm and 2 chromatids in the lower region Q arm, and each consisting of 2 sister chromatids condenses and become visible. Condensing in the sense they become thick, very short, and they can be seen very easily. Okay, so the microtubules are assembled in mitotic spindle. The microtubules are assembled in this for just catch holding the chromosomes near the centromeres. Nucleolus and nuclear envelope disappears. Why does the nuclear envelope and nucleolus will be disappears? Okay, so why? Because if the chromosomes are present inside the nucleus, how these tubular structures will be helping for the chromosomes to just divide process? They can't. Okay, so that is the reason here we can't observe the nuclear membrane and the nucleolus in the prophase. Okay, so let us go for the next phase. Second phase is called as a metaphase. Okay, so during this phase the chromosomes line up at the equator. If the cell is being just placed like this, so here we can observe the plate like structure. Each and every chromosome is adjusted, exactly they got together towards to the equatorial plate. Okay, so where we can observe the chromosomes are attached towards to the spindle fibers. The centromeres are helping for the chromosome to attach towards to the spindle fibers. Okay, the equatorial plate will be formed. Here we can observe the kinetochore. What is kinetochore? These are the kinetochores of the sister chromatids get joined towards to the spindle fibers. They provide the support towards to the centromeres to catch hold the each and every chromosome towards to the spindle fibers. Okay, so this kinetochore helps along with the centromeres to catch hold the chromosome towards to the spindle fibers. They helpful for equal distribution in the next phase, right? Next phase is the anaphase. This is the third phase. Centromere of each chromatid divides and chromatids are separated. Very clearly we can observe over here. Previous in the metaphase we have observed each and every chromosome with 4 chromatids. We have observed that they are been hanging freely with the help of a centromere, kinetochore and the spindle fibers. So here the centromeres along with the kinetochores, so they are ready to move towards to the polar regions, both the polar regions, each chromosome with 4 chromatids dividing 2 chromatids of P and Q. 2 chromatids of P and Q will be pulled by the spindle fibers and they will be moving towards to the opposite polar regions from the equatorial plate in the metaphase. The chromatids with their own centromeres are formed and are called as daughter chromosomes. After divided, they will become a daughter chromosome. So, movement of each chromosome occurs towards to their respective places. The spindle fibers, centromeres along with the kinetochores, so they are helpful for double set of the chromosomes, nothing but P and Q. P and Q. So, they were divided, they were closed in the metaphase in a equatorial plate, and when coming to the anaphase region, the centromere in the middle region will be, so the middle region, the center in the centromere will be helpful for pulling towards to the opposite poles for forming 2 daughter chromosomes. So, movement of chromosomes occur towards to their respective polar regions, upper region and the below region. Okay, and next fourth phase is the telophase. So, the changes are reversed to those of the prophase, exactly opposite towards to the prophase itself. So, here we can observe the despiralization, nucleolus and nuclear membrane will be reappears. So, there in the prophase it degenerates, it disappears. Nuclear membrane will be disappearing, but coming to this telophase region, we can observe the formation of a nuclear membrane and the nucleolus. So, here at the ultimate stage of the telophase, we can observe 2 daughter cells will be formed with equal number of chromosomes. Okay. Telophase and the cytokinesis. First we can observe the division of chromatids, division of the chromosomes, equal distribution, and then we can observe the cytoplasm will be divided along with the 2 nuclei. Okay, so nuclei, how we can observe the reappearance of the nucleus and the nuclear membrane will be formed in the telophase, and at the ending stage of the telophase we can observe division of the cytoplasm. Before to that, the division of nucleus occurs by the complete formation of nuclear membrane and the nucleolus inside to that each and every chromosome in an equal number will be maintained by both the cells, and then we can observe the first nucleus divides, karyokinesis, and next cytoplasm divides, cytokinesis, and completely dividing into 2 cells, 2 daughter cells. Right? And what is the cytokinesis? Already we have discussed. Here the cell is ready to divide into the equal number of chromosomes. So, here we can observe the phragmoplast. In the plant cells we can observe the phragmoplast. Okay, so what is a phragmoplast? So, it is responsible for maintaining as a plate. The process of division of cytoplasm, the spindle fragments gather at the equator region of the cell and form as a barrel. Barrel in the sense separating. Okay, so the ending regions, edges of two membrane regions. Okay, so two membranes will be formed in the two daughter cells. So cell plate is formed by the Golgi complex, and growth of cell plate is the centrifugal. What is centrifugal? The force of attraction will be away. From the center it goes away towards to the outer region. Okay, so for that the spindle fibers will be moving apart from each other to form a simple cells, nothing but separate cells, mother cell and the daughter cell. Again they will be undergoing the continuous division process. Okay, right. So now let us see what is the significance of mitosis. Already we have discussed at the starting part. So mitosis is an equational division, and it is a division through which daughter cells that are identical, same copies will be formed by the parental cell towards the daughter cell, and the daughter cell have the same amount and type of genetic constitution as that of the parental cell. The quality will be same, the characters will be same, and the type of chromosomes, genetic material will be maintained by the both parental cell and also the daughter cell. So this is the importance of the mitotic cell division. So clearly picture is given here. You see the parental cell, color of the chromosomes, you see one is bluish in color and one more is reddish in color. So as they both formed as a individuals, so again they are undergoing the division process, and here the blue and red also formed in both the cells. Yes, so same kind of characters will be distributed towards to the offsprings, and same number, quantity of the chromosomes also will be distributed towards to the daughter cells. Okay, so this is the importance, main significance of the mitotic cell division. Yes, thank you so much, dears.